Welcome to Midnight Anthropology. Tonight, we're exploring pharmacology. It's a journey into how tiny chemical compounds can unlock doors throughout your body, binding to receptors like keys fitting into locks, changing the very nature of how your cells communicate and function. Before we start, if you like these gentle explainer videos for sleep, I'd appreciate if you'd like and subscribe. Now, take a deep breath and find a comfortable spot. Let's begin. Pharmacology sits at the intersection of chemistry and life itself. It's the study of how chemical substances interact with living systems, how they alter the fundamental processes that keep us alive, and how we've learned to harness these interactions to treat disease, manage pain, and extend human life. The word comes from the Greek pharmakon, meaning drug or poison, and logos, meaning study. This dual nature, embedded in the very etymology, reminds us that the same substance can heal or harm depending on dose, context, and application. At its core, pharmacology asks two fundamental questions. First, what does the body do to a drug? A field called pharmacokinetics. This encompasses the journey of a molecule from the moment it enters your system until it leaves, tracking how it's absorbed through tissues, distributed via the bloodstream, chemically transformed by enzymes, and eventually eliminated from the body. Second, what does the drug do to the body, which we call pharmacodynamics? This explores the mechanisms of drug action, how molecules bind to specific targets, how they trigger cascades of cellular events, and ultimately, how they produce the effects we observe, whether therapeutic or adverse. These two branches of pharmacology are inseparable. A drug might have extraordinary potential to bind to its target receptor, but if it's immediately destroyed by stomach acid before it can be absorbed, that potential means nothing. Conversely, a drug might survive the journey through the digestive system perfectly intact, but if it cannot cross biological membranes to reach its site of action, it remains therapeutically useless. The art and science of pharmacology lies in understanding both aspects, in designing molecules that can survive the body's defenses, while still maintaining the precise chemical structure needed to produce healing effects. Modern pharmacology emerged from centuries of trial and error, from ancient healers who discovered that willow bark could ease pain, to medieval apothecaries who prepared tinctures and elixirs, to 19th century chemists who first isolated pure compounds from plant materials. The extraction of morphine from opium poppies in the early 1800s marked a turning point, demonstrating that specific molecules were responsible for therapeutic effects. This realization transformed medicine from an art based on tradition into a science grounded in chemistry and biology. Today, pharmacologists work with tools that would have seemed like magic to earlier generations, using computer modeling to predict how drugs will behave, genetic engineering to produce therapeutic proteins, and sophisticated analytical techniques to measure drug concentrations in blood plasma down to parts per billion. The scope of pharmacology extends far beyond simply treating disease. It encompasses understanding how drugs affect normal physiological processes, how genetic variations between individuals influence drug responses, how different drugs interact when taken together, and how drug effects change across the lifespan, from infancy to old age. Pharmacologists study not just medicines but also toxins, environmental chemicals, and substances of abuse, seeking to understand the full spectrum of how chemical compounds affect living organisms. The journey of a drug through your body begins the moment it enters your system, and that very first step, absorption, determines whether the drug will ever have a chance to exert its therapeutic effects. Absorption is the process by which a drug moves from its site of administration into the bloodstream, crossing biological barriers and navigating the complex terrain of human tissues. This seemingly simple movement involves overcoming substantial obstacles, and the route a drug takes profoundly influences its fate. Consider a tablet you swallow. That small disc of compressed powder must first disintegrate in the acidic environment of your stomach, releasing its active ingredient into the digestive fluids. The drug molecules must then dissolve in these fluids, a process influenced by the chemical properties of the molecule itself. Drugs that are highly water-soluble dissolve readily in the aqueous environment of the stomach and intestines, while drugs that are more fat-soluble may struggle to dissolve, potentially passing through the digestive system without ever being absorbed. Pharmaceutical scientists spend considerable effort formulating drugs to optimize this dissolution process using specialized coatings, particle sizes, and chemical forms to enhance solubility. Once dissolved, the drug faces its next challenge, crossing the intestinal wall to enter the bloodstream. The intestines are not simply passive tubes through which substances flow. They're lined with a single layer of epithelial cells, each cell packed tightly against its neighbors, creating a selective barrier between the contents of your gut and your internal circulation. Small, fat-soluble molecules can slip between these cells or pass directly through their lipid membranes by passive diffusion, moving from areas of high concentration to areas of low concentration without requiring any energy expenditure. Larger molecules, or those that are water-soluble, often cannot cross these membranes passively and must rely on specialized transport proteins embedded in the cell membranes, proteins that actively carry specific molecules across the barrier. The small intestine is remarkably well designed for absorption. Its inner surface is not smooth, but folded into countless tiny projections called villi, each villi covered with even smaller projections called microvilli, creating a surface area roughly equivalent to a tennis court, compressed into the space of your abdomen. This vast surface area maximizes the opportunity for drug molecules to encounter and cross the intestinal barrier. Blood vessels run through each villus, ready to carry absorbed drugs away toward the liver. But this is where many orally administered drugs encounter a crucial limitation known as the first pass effect. Blood from the intestines doesn't flow directly into general circulation. Instead, it travels first through the hepatic portal vein to the liver, an organ that acts as the body's primary chemical processing plant. The liver is packed with enzymes specifically designed to break down foreign substances, and many drugs are partially or even completely metabolized during this first pass through hepatic tissue. A drug might be extensively absorbed from the intestines, yet only a fraction of that absorbed dose reaches the systemic circulation because the liver has already begun breaking it down. This is why some drugs must be given in much higher oral doses than would be needed if they were administered directly into the bloodstream. The concept of bioavailability captures this reality. Bioavailability is defined as the fraction of an administered dose that reaches the systemic circulation unchanged. For a drug given intravenously, bioavailability is 100% by definition, since the drug is placed directly into the bloodstream. For oral drugs, bioavailability can range from nearly 100% for drugs that are well absorbed and resistant to first-pass metabolism, down to just a few percent for drugs that are poorly absorbed or extensively metabolized by the liver. Different routes of administration bypass some of these challenges. Drugs absorbed through the mucous membranes under the tongue, known as sublingual administration, enter blood vessels that drain directly into the superior vena cava, avoiding the first pass effect entirely. Drugs applied to the skin as patches can slowly diffuse through the layers of the epidermis and dermis to reach capillaries beneath, providing steady absorption over hours or days. Drugs inhaled into the lungs encounter an absorption surface even larger than that of the intestines, with the added advantage of extremely thin barriers between air and blood, allowing rapid entry into circulation. Injectable drugs can be placed subcutaneously just under the skin for gradual absorption, intramuscularly into muscle tissue for moderate absorption rates, or intravenously directly into veins for immediate effect. Each route has its advantages and limitations, its appropriate applications and contraindications. The choice depends not just on the chemical properties of the drug, but on the urgency of the clinical situation, the patient's ability to swallow or tolerate injections, the desired duration of effect, and the need to avoid or exploit first-pass metabolism. Once a drug enters the bloodstream, it begins a journey through a vast network of vessels that reaches every tissue in your body, a process we call distribution. But this is not simply a matter of drugs floating freely through blood vessels until they happen upon their target. Distribution is a complex interplay of blood flow, chemical properties, and biological barriers that determines which tissues receive high drug concentrations and which receive little or none. Blood flow is the primary driver of distribution. Tissues with rich blood supplies, organs like the brain, heart, liver, and kidneys, receive drug molecules quickly after absorption. These highly perfused organs can reach peak drug concentrations within minutes of intravenous administration. In contrast, tissues with sparser blood supplies, such as fat, bone, and resting skeletal muscle, receive drugs much more slowly, potentially taking hours to equilibrate with blood concentrations. But blood flow alone doesn't tell the complete story. A drug's chemical properties profoundly influence where it can go. The human body is essentially a collection of water-filled compartments separated by lipid barriers. Cell membranes, made primarily of fatty molecules arranged in double layers, surround every cell. To move from blood into tissues, drugs must cross these membranes. Fat-soluble drugs, also called lipophilic drugs, dissolve readily in these fatty barriers and can cross cell membranes easily, distributing widely throughout the body, including into the brain and other tissues protected by particularly tight barriers. Water-soluble drugs, or hydrophilic drugs, struggle to cross lipid membranes and tend to remain in the aqueous compartments of blood and interstitial fluid. Many drugs don't travel through the bloodstream as free molecules. Instead, they bind to proteins dissolved in the blood plasma, particularly a protein called albumin. This protein binding can be quite extensive, with some drugs more than 95% bound to plasma proteins at any given moment. Only the unbound fraction of a drug is free to cross membranes and exert pharmacological effects. The bound fraction acts as a reservoir, slowly releasing drug as the free fraction is metabolized or excreted, helping to maintain steady drug levels over time. However, this binding can also create complications. If two drugs compete for the same binding sites on albumin, one drug can displace the other, suddenly increasing the free concentration and potentially causing toxicity. Certain tissues in the body are protected by particularly stringent barriers. The blood-brain barrier is perhaps the most famous of these. The capillaries that supply the brain are fundamentally different from capillaries elsewhere in the body. Their endothelial cells are joined by extremely tight junctions, leaving virtually no gaps between cells. Surrounding these capillaries are the foot processes of astrocytes, specialized brain cells that provide an additional layer of selectivity. Together, these features create a barrier that allows essential nutrients like glucose to pass, while blocking most drugs and potentially harmful substances. Only small lipophilic molecules can readily cross the blood-brain barrier by passive diffusion. This protective mechanism is wonderful for keeping toxins away from delicate neural tissue, but it poses significant challenges for treating brain disorders. Many potentially useful drugs for neurological diseases cannot reach their targets because they cannot cross this barrier. Similar barriers protect other sensitive organs. The blood-testis barrier shields developing sperm cells from potentially mutagenic compounds. The placental barrier, though less restrictive than once believed, still provides some protection for the developing fetus. Understanding these barriers is crucial for drug design and for predicting which drugs might cause adverse effects in protected tissues. The volume of distribution is a pharmacokinetic parameter that captures the apparent space into which a drug distributes throughout the body. A drug that remains mostly in the bloodstream will have a small volume of distribution, close to the actual volume of blood plasma. A drug that extensively penetrates tissues will have a large volume of distribution, potentially many times larger than the total volume of body water, reflecting the fact that tissue concentrations exceed blood concentrations due to binding or accumulation in specific compartments. As drugs circulate through your bloodstream and distribute into tissues, your body begins the process of chemical transformation, converting these foreign molecules into forms that can be more easily eliminated. This process, called drug metabolism or biotransformation, occurs primarily in the liver. The liver is uniquely suited for this role. It receives blood from both the hepatic artery, carrying oxygenated blood, and from the hepatic portal vein, carrying nutrient-rich and drug-laden blood from the intestines. Liver cells, called hepatocytes, are packed with enzymes dedicated to chemical transformation. Drug metabolism typically occurs in two phases. Phase 1 reactions introduce or expose a functional group on the drug molecule, usually making it slightly more polar and water-soluble. The most important Phase 1 reactions are performed by the cytochrome P450 family of enzymes, often abbreviated CYP450. These are remarkable molecular machines, each containing an iron atom at their active site that can insert an oxygen atom into otherwise stable chemical bonds. The human genome encodes dozens of different CYP450 enzymes, though many drugs are processed by just a handful, particularly CYP3A4, CYP2D6, CYP2C9, and CYP2C19. These enzymes catalyze oxidation reactions, adding oxygen atoms to drugs in the form of hydroxyl groups. This seemingly minor chemical modification can dramatically change a drug's properties. The added oxygen makes the molecule more polar, more water-soluble, and often less able to cross cell membranes. Sometimes this oxidation directly inactivates the drug. Other times, the oxidation is just the first step, creating a molecular handle that Phase 2 enzymes can grab onto. Phase 2 reactions involve conjugation, the attachment of large polar molecules to the drug, or its Phase 1 metabolite. Common conjugation reactions include glucuronidation, the addition of glucuronic acid, and sulfation, the addition of a sulfate group. The conjugated products are typically much larger and more water-soluble than the original drug, making them ideal candidates for excretion. Not all metabolism inactivates drugs. Some drugs are administered as inactive prodrugs that require metabolic activation. Codeine, for example, must be converted by CYP2D6 into morphine to exert its analgesic effects. Other drugs produce toxic metabolites. Acetaminophen is normally safely metabolized through glucuronidation and sulfation, but a small fraction is converted into a toxic metabolite called NAPQI. At normal doses, this toxic metabolite is immediately neutralized by glutathione, but in overdose situations, glutathione stores become depleted, and NAPQI accumulates, causing potentially fatal liver damage. The activity of metabolic enzymes varies considerably between individuals. Genetic differences can result in people having too many copies of a particular enzyme, making them ultra-rapid metabolizers who break down certain drugs so quickly that standard doses don't work. Others may have non-functional versions of enzymes, making them poor metabolizers who accumulate drugs to toxic levels at normal doses. Perhaps most importantly for clinical practice, drugs can interact through their effects on metabolic enzymes. Some drugs are enzyme inducers, meaning they increase the production of certain CYP450 enzymes when taken regularly. A patient taking an enzyme inducer will metabolize other drugs more quickly than expected, potentially reducing their effectiveness. Other drugs are enzyme inhibitors, blocking the activity of metabolic enzymes and causing other drugs to accumulate. When a patient takes an enzyme inhibitor along with another drug, metabolized by the same enzyme, drug concentrations can rise dramatically, sometimes to toxic levels. Everything that enters your body must eventually leave, and the process by which drugs and their metabolites exit the system is called excretion. While several organs participate in drug elimination, the kidneys bear primary responsibility for this crucial function, filtering blood and selectively removing waste products and foreign chemicals. Your kidneys are extraordinary organs, each about the size of your fist, containing roughly 1 million functional units called nephrons. Each nephron consists of a glomerulus, a tangled ball of capillaries, connected to a long tubule that winds through the kidney tissue. Blood enters the glomerulus under pressure, and this pressure forces water and small molecules, including drug molecules, through the capillary walls into the tubule. This process, glomerular filtration, is remarkably efficient, filtering your entire blood volume multiple times each day. However, filtration alone would be wasteful, removing not just drugs and waste products, but also valuable nutrients, electrolytes, and water. As the filtered fluid flows through the tubule, the kidney performs selective reabsorption, pulling useful substances back into the bloodstream while leaving waste products behind. This is where a drug's chemical properties again become crucial. Small, water-soluble molecules remain in the tubular fluid and are carried away in urine. Fat-soluble molecules can be reabsorbed across the tubular membrane back into the bloodstream, potentially recycling back through the body multiple times, before eventually being metabolized into more water-soluble forms that can be excreted. The kidney tubules also perform active secretion, using specialized transport proteins to actively pump certain drugs from blood into tubular fluid. This secretion can remove drugs that are highly bound to plasma proteins, drugs that couldn't be removed efficiently by filtration alone. The combination of filtration, reabsorption, and secretion determines the overall renal clearance of a drug. Kidney function varies with age and disease. Newborns have immature kidneys with reduced filtration capacity. Kidney function gradually improves through the first few years of life, then remains relatively stable through adulthood, before gradually declining with age. Many elderly individuals have significantly reduced kidney function compared to young adults, even in the absence of kidney disease. This age-related decline, combined with the prevalence of conditions like diabetes and hypertension that can damage kidneys, means that dose adjustments are frequently necessary in older patients to prevent drug accumulation. Some drugs and metabolites are excreted through bile, a digestive fluid produced by the liver. Bile flows into the intestines, where it aids in fat digestion. Drugs excreted in bile may be eliminated in feces, or they may be reabsorbed from the intestines back into the bloodstream, a process called enterohepatic recirculation. This recirculation can prolong drug effects, as the drug cycles repeatedly between liver and intestines. Other routes of excretion include the lungs, particularly for volatile substances and gases, sweat glands, saliva, and breast milk. While these routes typically account for only minor fractions of total drug elimination, excretion in breast milk deserves special attention because it can expose nursing infants to maternal medications. The concept of half-life ties together all aspects of pharmacokinetics. A drug's half-life is the time required for the blood concentration to decrease by 50%. This single number reflects the combined effects of distribution, metabolism, and excretion. A drug with a short half-life is rapidly eliminated, requiring frequent dosing to maintain therapeutic levels. A drug with a long half-life persists in the body, allowing less frequent dosing, but also meaning that it takes longer to reach steady-state concentrations when therapy begins, and longer to be completely eliminated after therapy stops. Understanding half-life helps predict how often a drug must be dosed. Generally, a drug is given at intervals equal to or shorter than its half-life. It also takes approximately 5 half-lives for a drug to reach steady-state concentrations during chronic dosing, and 5 half-lives to be essentially eliminated after dosing stops. A drug with a half-life of 6 hours reaches steady state in about 30 hours of regular dosing, while a drug with a half-life of 24 hours takes about 5 days. Having explored how drugs move through the body, we now turn to how they actually work, to the molecular mechanisms through which chemical compounds alter cellular function. The vast majority of drugs exert their effects by binding to specific proteins called receptors. These receptors evolved to respond to endogenous molecules, the body's own chemical messengers like hormones and neurotransmitters. Drugs work by mimicking, blocking, or modifying the effects of these natural messengers. The relationship between a drug and its receptor is often described using the lock and key metaphor. Each receptor has a binding site, a 3-dimensional pocket with a specific shape and distribution of chemical properties. A drug that fits this binding site can bind to the receptor. This binding is usually reversible, held together by weak chemical forces, including hydrogen bonds and electrostatic attractions. The strength of this binding relationship is described by affinity. A drug with high affinity binds tightly to its receptor, meaning that even at low concentrations, a significant fraction of receptors will be occupied. A drug with low affinity binds weakly, requiring higher concentrations. But binding alone doesn't necessarily produce an effect. For a drug to produce a pharmacological effect after binding, it must also have what we call intrinsic activity or efficacy, the ability to activate the receptor and trigger downstream signaling events. There are 4 major families of receptors in the human body. Ion channel receptors are protein complexes that span the cell membrane, forming a pore or channel through which ions can flow. When a neurotransmitter or drug binds to the receptor, it causes a conformational change that opens the channel, allowing ions like sodium, potassium, calcium, or chloride to flow across the membrane. This ion flow changes the electrical properties of the cell. These receptors produce very rapid effects, typically within milliseconds. G-protein-coupled receptors, or GPCRS, are the largest family of receptors and the target of approximately 30% of all currently approved drugs. GPCRS don't directly alter ion flow or enzyme activity. Instead, they work through intermediary proteins called G proteins that can trigger complex cascades of intracellular events. Enzyme-linked receptors have an extracellular domain that binds signaling molecules and an intracellular domain that has enzymatic activity. When a ligand binds to the extracellular side, it activates the enzymatic activity on the intracellular side. Receptor tyrosine kinases are important members of this family, activated by growth factors and hormones like insulin. Intracellular receptors are located inside cells, either in the cytoplasm or nucleus. Their ligands must be sufficiently lipophilic to cross the cell membrane. When a ligand binds to an intracellular receptor, the receptor-ligand complex typically moves to the nucleus, where it binds to specific DNA sequences, directly regulating gene transcription. Steroid hormones like cortisol, estrogen, and testosterone work through this mechanism. The effects typically take hours to days to fully develop. Among the 4 major receptor families, G-protein coupled receptors deserve special attention due to their central role in pharmacology and their remarkable versatility. These receptors are involved in nearly every physiological process, from neurotransmission to hormone action, from sensory perception to immune responses. Understanding how GPCRS work provides insight into how a vast array of medications produce their therapeutic effects. The basic structure of a GPCR is distinctive and highly conserved across the approximately 800 different GPCRS encoded in the human genome. Each receptor consists of a single protein chain that winds back and forth across the cell membrane 7 times, creating 7 transmembrane helices. These helices are connected by loops that extend into the extracellular space and into the cytoplasm. The extracellular portions form the binding site for ligands, while the intracellular portions interact with G proteins. G proteins, the partners that give these receptors their name, are heterotrimeric proteins made of 3 subunits called alpha, beta, and gamma. When a GPCR is inactive, an associated G protein sits nearby with GDP, guanosine diphosphate, bound to its alpha subunit. When a ligand binds to the GPCR, it causes a conformational change in the receptor that allows it to interact with the G protein, catalyzing the exchange of GDP for GTP, guanosine triphosphate. This exchange activates the G protein, causing the alpha subunit to separate from the beta-gamma complex. Both the free alpha subunit and the beta-gamma dimer can then go on to interact with other proteins, triggering downstream effects. There are several different types of G-alpha subunits, and which type a particular GPCR couples determines what effects its activation produces. G-alpha-OS, the stimulatory type, activates the enzyme adenylyl cyclase, which produces cyclic AMP, a second messenger that activates protein kinase A and triggers a cascade of phosphorylation events. Many hormones work through this pathway. Epinephrine binding to beta-adrenergic receptors activates G-alpha-S, leading to increased heart rate and force of contraction, among many other effects. G-alpha-I, the inhibitory type, does the opposite, inhibiting adenylyl cyclase and reducing cyclic AMP levels. Some opioid receptors couple to G-alpha-I, contributing to their pain-relieving effects. G-alpha-Q activates phospholipase C, an enzyme that cleaves a membrane phospholipid into 2 second messengers, IP3 and DAG, which respectively trigger calcium release from intracellular stores and activate protein kinase C. Many hormones that cause smooth muscle contraction work through this pathway. This system provides remarkable signal amplification. A single activated GPCR can activate multiple G proteins. Each activated G protein can activate multiple enzyme molecules. Each enzyme can generate many second messenger molecules. Each second messenger can activate multiple kinase molecules. Each kinase can phosphorylate multiple target proteins. Through this cascade, the binding of a few hormone molecules to receptors on a cell surface can trigger massive changes in cellular function. The system also includes mechanisms for desensitization. When a GPCR is stimulated for prolonged periods, it can be phosphorylated by specialized kinases, which creates binding sites for proteins called beta-arrestins. These both block further G-protein coupling and target the receptor for internalization into the cell, temporarily removing it from the cell surface. This prevents excessive stimulation and allows the cell to adapt to sustained signals. Many important drugs target GPCRS. Beta blockers, used to treat high blood pressure and heart disease, block beta-adrenergic receptors, preventing epinephrine and norepinephrine from activating them. Antihistamines block histamine receptors, preventing allergic responses. Many antipsychotic and antidepressant drugs modulate dopamine or serotonin receptors. Opioid pain medications activate mu-opioid receptors. The list encompasses drugs for asthma, migraine, nausea, glaucoma, and countless other conditions. With an understanding of how drugs bind to receptors, we can now explore the different ways drugs can modulate receptor activity. The terminology here is precise and important, distinguishing between drugs that activate receptors, drugs that block them, and the various intermediate possibilities. An agonist is a drug that binds to a receptor and activates it, producing a response similar to the natural ligand. A full agonist can produce the maximum possible response that the receptor system can generate, fully activating the receptor when enough agonist molecules are bound. Think of adrenaline binding to beta-adrenergic receptors during a stress response, or morphine binding to opioid receptors to relieve pain. These molecules fit into the receptor's binding site and trigger the conformational changes that lead to G protein activation or channel opening. A partial agonist also binds to the receptor and activates it, but even when all receptors are occupied by the partial agonist, it cannot produce the maximal response. Partial agonists have lower intrinsic activity than full agonists. Interestingly, in a system where a full agonist is already present, adding a partial agonist can actually reduce the response. Because the partial agonist competes with the full agonist for binding sites, but produces less activation per bound receptor. Buprenorphine, used in treating opioid addiction, is a partial agonist at mu-opioid receptors. It provides enough receptor activation to prevent withdrawal symptoms, but not enough to produce the full euphoric effects of drugs like heroin. An antagonist binds to a receptor but produces no activation. It simply occupies the binding site, preventing agonists from binding and producing their effects. Antagonists are blocking drugs. They don't initiate responses, they prevent responses. Competitive antagonists bind to the same site as the agonist. Because binding is reversible, an agonist and a competitive antagonist are literally in competition for the receptor. If you increase the concentration of agonist high enough, you can overcome the blockade, displacing the antagonist from the receptors. Many therapeutic antagonists work this way. Naloxone, used to reverse opioid overdoses, is a competitive antagonist at opioid receptors. It has higher affinity than most opioid drugs, so when given, it rapidly displaces them from receptors, reversing their effects. Non-competitive antagonists bind to a different site on the receptor, or they bind irreversibly. They reduce the maximal response that can be achieved, regardless of how much agonist is present. Some poison gases are irreversible antagonists at acetylcholine receptors, permanently inactivating them. Beyond these direct effects on single receptors, drugs can interact in ways that affect their combined activity. When two drugs that produce similar effects are given together, they may show additive effects, where the total response equals the sum of their individual effects. In some cases, drugs show synergistic effects, where the combined effect is greater than the sum of individual effects. This can be therapeutically useful, allowing lower doses of each drug to achieve the desired effect, potentially reducing side effects. However, synergy can also be dangerous if it's unexpected, leading to excessive effects. Drug-drug interactions can also occur through pharmacokinetic mechanisms, as we discussed during metabolism. When one drug inhibits the enzyme that metabolizes another drug, the second drug accumulates to higher concentrations than expected. This is effectively a pharmacokinetic enhancement of effect. Conversely, enzyme induction can reduce drug concentrations and effectiveness. Some drugs are functional antagonists, producing opposite effects through different mechanisms. A drug that causes bronchodilation by activating beta receptors in the lungs functionally opposes a drug that causes bronchoconstriction, even though they work through completely different receptor systems. One of the fundamental relationships in pharmacology is between the dose of a drug and the response it produces. This relationship, when plotted on a graph, creates what we call a dose-response curve, and understanding these curves provides crucial insights into how drugs work and how to use them safely and effectively. If you give increasing doses of a drug and measure the response at each dose, you typically see a characteristic pattern. At very low doses, there may be no detectable effect. As the dose increases, a response begins to appear. Further dose increases produce larger responses, but the relationship isn't always linear. Eventually, you reach doses where further increases produce little additional effect. The curve plateaus, reaching a maximum response. When plotted with drug concentration on the x-axis and response on the y-axis, this creates a curve that typically has a sigmoidal or S-shaped form, especially when the x-axis uses a logarithmic scale. The curve starts flat at low doses, rises steeply through the middle range, and flattens again at high doses. Two key parameters characterize these dose-response curves. The first is potency, which refers to the dose required to produce a particular level of effect. A potent drug produces its effects at low doses. A less potent drug requires higher doses for the same effect. Potency is often quantified as the ED50, the effective dose that produces 50% of the maximal response. On a dose-response curve, a more potent drug's curve is shifted to the left, reaching the same response levels at lower doses. Potency is determined primarily by affinity, how tightly the drug binds to its receptor. A drug with higher affinity requires fewer drug molecules to occupy enough receptors to produce an effect. However, potency alone doesn't determine a drug's therapeutic utility. A drug could be very potent but impractical to use if it has a short duration of action, poor bioavailability, or serious side effects. The second parameter is efficacy, the maximum effect a drug can produce. On the dose-response curve, efficacy is represented by the height of the plateau, the ceiling of the response. Efficacy is determined by intrinsic activity, how effectively the drug activates receptors once bound. Two drugs binding to the same receptor might have very different efficacies. A full agonist has high efficacy, producing the maximum possible response. A partial agonist has lower efficacy, unable to produce the full response no matter how high the dose. In clinical practice, efficacy is often more important than potency. A less potent drug that produces a strong therapeutic effect may be preferable to a highly potent drug that produces only weak effects. The difference between a drug that reduces blood pressure by 5 millimeters of mercury and one that reduces it by 20 millimeters matters far more than whether you need to take 10 milligrams or 50 milligrams to achieve that effect. The slope of the dose-response curve in its middle section also carries meaning. A steep slope indicates that small changes in dose produce large changes in effect, requiring careful dose titration. A shallow slope suggests that dose adjustments within a broad range may produce similar effects. For all their benefits, drugs can also cause harm, and understanding the relationship between therapeutic and toxic effects is fundamental to safe pharmacology. This relationship is quantified through the therapeutic index. The therapeutic index is calculated as the ratio of the toxic dose to the effective dose. In animal studies, this is often expressed as LD50 divided by ED50, where LD50 is the lethal dose that kills 50% of test animals, and ED50 is the effective dose that produces the desired therapeutic effect in 50%. A high therapeutic index indicates a wide margin of safety. Even if a patient receives several times the therapeutic dose, serious toxicity is unlikely. Penicillin antibiotics have very high therapeutic indices because they target bacterial cell wall synthesis, a process that doesn't occur in human cells. A low therapeutic index indicates a narrow margin of safety. Small increases above the therapeutic dose can produce toxicity. Drugs with narrow therapeutic indices require careful dose monitoring. Warfarin, an anticoagulant, has a narrow therapeutic index. Too little and it won't prevent blood clots. Too much and it causes dangerous bleeding. Lithium, used in bipolar disorder, and digoxin for heart failure, both have narrow therapeutic indices requiring careful management. The therapeutic window or therapeutic range describes the range of drug concentrations in blood that produces therapeutic effects in most patients without causing toxicity. For drugs with wide therapeutic indices, this window is broad and forgiving. For drugs with narrow indices, the window may be quite small. Individual variation adds complexity to these concepts. The ED50 and TD50 are population averages, but individual patients may be much more or less sensitive than average. Factors affecting this individual variation include genetics, particularly genetic polymorphisms in drug metabolizing enzymes or drug receptors, and age, with both very young and very old patients often being more sensitive to drugs. Drugs can be organized and classified in multiple ways. The most clinically useful classification is by therapeutic use, grouping drugs by what they're used to treat. Analgesics are drugs that relieve pain. This broad category includes non-opioid analgesics like acetaminophen and non-steroidal anti-inflammatory drugs, the NSAIDs, which work peripherally, reducing inflammation and blocking pain signals at their source. Aspirin, ibuprofen, and naproxen are familiar NSAIDs that inhibit cyclooxygenase enzymes, reducing production of prostaglandins that mediate inflammation and pain. Opioid analgesics like morphine, oxycodone, and fentanyl work centrally in the brain and spinal cord, activating opioid receptors that modulate pain perception. Antibiotics treat bacterial infections by killing bacteria or preventing their growth. Different classes target different aspects of bacterial biology. Beta-lactam antibiotics, like penicillins and cephalosporins, interfere with bacterial cell wall synthesis. Tetracyclines and macrolides inhibit bacterial protein synthesis. Fluoroquinolones interfere with bacterial DNA replication. Cardiovascular drugs form a large and diverse category. Antihypertensives lower blood pressure through various mechanisms. Beta blockers like metoprolol block beta-adrenergic receptors in the heart, reducing heart rate and contractility. ACE inhibitors like lisinopril block the enzyme that produces angiotensin II, a hormone that constricts blood vessels. Calcium channel blockers like amlodipine prevent calcium entry into vascular smooth muscle cells, causing relaxation and dilation. Other cardiovascular drugs treat heart failure, abnormal heart rhythms, high cholesterol, and clotting disorders. Statins like atorvastatin inhibit the enzyme HMGCOA reductase, reducing cholesterol synthesis in the liver. Drugs affecting the central nervous system include antidepressants, antipsychotics, and anxiolytics. Selective serotonin reuptake inhibitors, the SSRIs, like fluoxetine and sertraline, block the reuptake of serotonin from synapses, increasing its availability. This helps alleviate depression and anxiety. Antipsychotic drugs primarily block dopamine receptors. Benzodiazepines like diazepam enhance GABA-mediated inhibition in the brain, providing anxiolytic and sedative effects. Drugs for diabetes management include insulin and various oral medications. Metformin reduces glucose production by the liver and improves insulin sensitivity. Sulfonylureas stimulate insulin release from pancreatic beta cells. To understand how many drugs work, particularly those affecting mood, behavior, and neurological function, we must understand neurotransmitters, the chemical messengers that neurons use to communicate with each other. Neurotransmitters are small molecules synthesized within neurons, stored in tiny membrane-bound vesicles, and released into the narrow gap between neurons called the synapse. When an electrical signal travels down a neuron and reaches the axon terminal, it triggers the opening of voltage-gated calcium channels. Calcium rushing into the terminal causes vesicles to fuse with the cell membrane and release their neurotransmitter contents into the synaptic cleft. These neurotransmitter molecules diffuse across the narrow gap, typically only about 20 nanometers wide, and bind to receptors on the postsynaptic neuron, triggering responses that can either excite or inhibit the receiving neuron. The human brain uses dozens of different neurotransmitters, but we'll focus on two that are particularly important pharmacological targets: dopamine and serotonin. Dopamine is a catecholamine neurotransmitter involved in movement control, reward and motivation, and cognitive function. It's synthesized from the amino acid tyrosine through a series of enzymatic steps. First, tyrosine hydroxylase converts tyrosine to L-dopa. Then aromatic amino acid decarboxylase converts L-dopa to dopamine. Dopaminergic neurons, neurons that produce and release dopamine, are concentrated in specific brain regions, including the substantia nigra, which projects to the striatum and controls movement, and the ventral tegmental area, which projects to the nucleus accumbens and prefrontal cortex, and is involved in reward and motivation. Dopamine's role in movement becomes starkly clear in Parkinson's disease, where the progressive death of dopamine-producing neurons in the substantia nigra leads to the characteristic tremor, rigidity, bradykinesia, or slowness of movement, and difficulty initiating movement. The treatment approach exploits the dopamine synthesis pathway. Dopamine itself cannot cross the blood-brain barrier, but its precursor L-dopa can. Treatment with levodopa allows remaining dopaminergic neurons to produce more dopamine, helping restore dopamine levels and improve motor symptoms. Levodopa is typically combined with carbidopa, which blocks the enzyme that converts levodopa to dopamine in the periphery, ensuring more levodopa reaches the brain. Dopamine is also central to the brain's reward system, the circuitry that evolved to reinforce behaviors essential for survival, like eating and reproduction. When we experience something pleasurable or rewarding, dopamine neurons fire, releasing dopamine in regions including the nucleus accumbens. This dopamine signal reinforces behaviors that led to the reward, making us more likely to repeat them. Drugs of abuse hijack this system in various ways. Cocaine blocks the dopamine transporter, the protein that normally removes dopamine from the synaptic cleft, and pumps it back into the presynaptic neuron. By blocking this reuptake, cocaine prevents dopamine clearance and causes it to accumulate in synapses. The resulting surge of dopamine produces intense euphoria, but also drives addiction, as the brain's reward system becomes increasingly sensitized to the drug. Schizophrenia has been linked to excessive dopamine activity in certain brain pathways, particularly the mesolimbic pathway. The dopamine hypothesis of schizophrenia suggests that positive symptoms like hallucinations and delusions result from excessive dopamine transmission. Antipsychotic medications work primarily by blocking dopamine receptors, particularly D2 receptors, reducing the effects of excessive dopamine signaling. However, because dopamine pathways also control movement, blocking dopamine receptors throughout the brain can cause motor side effects resembling Parkinson's disease. Serotonin, chemically known as 5-hydroxytryptamine or 5-HT, is another monoamine neurotransmitter with widespread effects throughout the brain and body. It's synthesized from the amino acid tryptophan through a two-step process. While most of the body's serotonin is actually found in the gut, where it regulates intestinal movements and secretion, the serotonin in the brain plays crucial roles in mood regulation, sleep-wake cycles, appetite, pain perception, and many other functions. Serotonergic neurons are concentrated in the RAFE nuclei in the brainstem, but send projections throughout the brain. The serotonin hypothesis of depression suggests that reduced serotonin activity in certain brain regions contributes to depressive symptoms. This hypothesis emerged from observations that drugs affecting serotonin could influence mood. Selective serotonin reuptake inhibitors, SSRIs, like fluoxetine, sertraline, and escitalopram, block the serotonin transporter protein that normally removes serotonin from the synaptic cleft and returns it to the presynaptic neuron. By blocking this reuptake, SSRIs allow serotonin to remain in the synapse longer, enhancing serotonergic transmission. However, these drugs don't immediately cure depression. Instead, they require several weeks of treatment before full therapeutic effects emerge, suggesting that their benefits result not from the immediate increase in synaptic serotonin, but from adaptive changes in the brain that occur over time in response to elevated serotonin levels. Both dopamine and serotonin are cleared from synapses not just by reuptake transporters, but also by enzymatic breakdown. Monoamine oxidase, abbreviated MAO, is an enzyme found in mitochondria that breaks down both dopamine and serotonin, along with other monoamine neurotransmitters like norepinephrine. MAO inhibitors were among the first antidepressants discovered in the 1950s, working by preventing monoamine breakdown and thereby increasing neurotransmitter availability. However, They fell out of favor for routine use because they interact with tyramine, a substance found in aged cheeses, cured meats, and fermented foods, potentially causing dangerous spikes in blood pressure. The journey from discovering a promising molecule to having an approved medication available in pharmacies is long, expensive, and fraught with failure. The process typically takes 10 to 15 years and costs billions of dollars, with most candidate drugs failing somewhere along the way. Drug development typically begins with basic research, scientists studying disease mechanisms and identifying potential drug targets, whether receptors, enzymes, or other proteins whose activity could be modified to treat disease. Once a target is identified, researchers screen thousands or even millions of compounds, looking for molecules that interact with the target in promising ways. From thousands of compounds tested, perhaps a few dozen show enough promise to warrant further investigation. These hit compounds undergo medicinal chemistry optimization, where chemists systematically modify the molecular structure to improve potency, selectivity, and drug-like properties. These candidate compounds enter preclinical testing, which is conducted entirely in laboratory settings and animal models. Researchers test whether the compound actually produces the expected biological effects, what doses are needed, how it's absorbed and distributed, how it's metabolized, and whether it shows signs of toxicity. Preclinical testing is extensive and includes studies of acute toxicity from single doses, chronic toxicity from repeated dosing over weeks or months, effects on reproduction and fetal development, and potential to cause cancer. Compounds are tested in cell cultures and in at least 2 species of animals, typically a rodent like mice or rats and a non-rodent like dogs or monkeys. Only compounds that successfully navigate preclinical testing, demonstrating both efficacy and acceptable safety profiles in animals, advance to clinical trials in humans. Before even the first human receives the experimental drug, the developer must submit an investigational new drug application to the FDA, providing all preclinical data and detailed plans for the clinical studies. The FDA has 30 days to review the application and can place the study on hold if safety concerns arise. Clinical trials proceed through a series of phases, each with distinct goals and involving progressively larger numbers of participants. Phase 1 trials are the first tests in humans, typically involving 20 to 100 healthy volunteers. The primary goals are to assess safety, determine how the drug is absorbed, distributed, metabolized, and excreted in humans, and find the maximum tolerated dose. Phase 1 trials start with very small doses and gradually escalate, carefully monitoring participants for adverse effects. These trials usually last several months. Approximately 70% of drugs successfully complete Phase 1. Phase 2. Trials advance to testing the drug in people who actually have the disease or condition it's meant to treat. These trials involve 100 to 300 patients and focus on whether the drug actually works, providing preliminary evidence of efficacy. They also continue to monitor safety and begin to explore optimal dosing regimens. Phase 2. Trials may be divided into Phase 2A, which focuses on dosing, and Phase 2B, which provides more robust evidence of efficacy. These trials may last from several months to 2 years. Only about 1/3 of drugs entering Phase 2 successfully complete this stage. About 33% of drugs that enter Phase 2 successfully complete it and advance to Phase 3. These are large, expensive trials involving hundreds to thousands of patients at multiple medical centers across different geographic regions. Phase 3 trials are designed to definitively demonstrate efficacy compared to placebo or existing treatments, and to gather comprehensive safety data from diverse patient populations. These trials are typically randomized, meaning patients are randomly assigned to receive either the experimental drug or a controlled treatment, and double-blind, meaning neither patients nor doctors know who receives which treatment until after the trial concludes. This rigor helps eliminate bias and ensure reliable results. Phase 3 trials can last several years and may cost hundreds of millions of dollars. Only about 25 to 30% of drugs entering Phase 3 will ultimately gain FDA approval. If the results demonstrate that the drug is both safe and effective, the developer can submit a new drug application to the FDA. This application is massive, often containing hundreds of thousands of pages of data, including all preclinical studies, all clinical trial results, detailed information about the drug's chemistry and manufacturing, proposed labeling, and plans for post-marketing surveillance. The FDA then conducts a thorough review, which typically takes 6 to 12 months for a standard application, or 6 months for a priority review granted to drugs treating serious conditions where no adequate alternatives exist. A team of medical officers, chemists, pharmacologists, and statisticians examines all the data, looking for evidence of efficacy, characterizing the safety profile, and determining whether benefits outweigh risks. If approved, the drug can be marketed and prescribed, but surveillance doesn't end there. Phase 4, also called post-marketing surveillance, continues to monitor the drug's safety in much larger populations over longer periods than was possible in clinical trials. Rare adverse effects that occur in perhaps 1 in 10,000 people wouldn't be detected in trials of a few thousand participants. Sometimes serious problems only emerge after millions of people have taken a drug. The FDA can require additional studies as conditions of approval, and can withdraw approval if serious safety concerns emerge. The entire process from initial discovery to FDA approval typically takes 10 to 15 years and costs an estimated $2 to $3 billion when accounting for all the failed candidates along the way. Of every 5,000 compounds that enter preclinical testing, approximately 5 will make it to clinical trials, and only 1 will ultimately receive FDA approval. This high failure rate and long timeline explain why new drugs are expensive and why pharmaceutical development remains one of the riskiest and most costly endeavors in all of industry. From the first moment a drug enters your body to its final exit, from the molecular dance at receptor sites to the years of careful testing that bring medicines to your bedside, pharmacology remains one of humanity's most remarkable achievements. The science of healing written in the language of chemistry. Thanks for being here. Sleep well.